Welcome, everyone. Thank you for joining our presentation today titled Lateral Flow Manufacturing Strategies and Scale Up Towards Point of Care Testing. It is my pleasure to introduce to you Dr. Chris Fronczek, Application Scientist, BioDot Incorporated. For a complete biography on our speaker today, please visit the tab at the top of your screen. If any questions arise during the presentation, we encourage you to submit them in the Q&A box. Our speaker will address your questions following at the end of the presentation. Please join me in welcoming Dr. Chris Fronczek. I will now turn the presentation over to him.
Hello, I'm Chris and I'm an application scientist with BioDot. I'm going to be discussing lateral flow manufacturing strategies and scale up towards point of care testing. A little bit about BioDot. We are a 26-year-old privately held company. We supply systems and solutions to various marketplaces from lateral flow diagnostics to life sciences to clinical. We're very customer focused. We're collaborative in nature. We're very application focused. Our global headquarters is in Irvine, California, where all of our manufacturing and quality is done. Recently, we opened East Coast for the United States location in Hillsborough, North Carolina. We have European office in the UK, a Chinese office in Shanghai, as well as distributors worldwide. We focus on a variety of markets from point of care tests to cytogenetics, molecular diagnostics, and even medical and industrial. However, we really got started in lateral flow devices. Particularly in the last 8 months, we've been very heavily focused in manufacturing of COVID-19 tests. The lateral flow device manufacturing process can be subdivided in a couple of different categories. First is the deposition of reagent, what I will refer to as dispensing. This involves depositing antibody test and control lines on nitrocellulose membrane, treating any sample pads or wicking pads beforehand, and depositing gold conjugate on the conjugate pad. Next is drying of all layers, and then assembling the layers in a process called lamination before cutting and assembling. Lateral flow manufacturing can be subdivided into batch manufacturing and inline manufacturing. Batch manufacturing is smaller scale production from initial R&D to hundreds of thousands of devices per year. Inline manufacturing is higher throughput to millions of devices per year. And this talk will focus not only on each one, but the process of transitioning from batch to inline. When considering reagent deposition, there are several critical factors which must be looked at. First and foremost is dispense volume and accuracy. This is critical for the development of sensitive tests. Volumetric accuracy should be within plus or minus 3% of the desired target volume, with better than 5% drop-to-drop precision. Dispense position along the membrane should also be considered. Oftentimes, reagents must be within plus or minus 50 microns of the desired position. This is important to avoid reagent overlap, in addition to, for example, tester control line omission because of overlap. For these more challenging applications, vision systems or fiducial markings can be used. It's critical to avoid cross-contamination, particularly to avoid false positives in testing. This can be accomplished through non-contact dispensing. It's important to be able to work over a wide range of fluid rheology, from blocking buffers to gold conjugates to antibodies, in addition to some more viscous concentrations. Typically, this process will be over a wide dynamic range from nanoliters to microliters per drop of reagent. However, newer applications, including high-density microarrays in lateral flow, often require picoliter volumes. Dispensing technology should be scalable from hundreds of thousands to tens of millions of tests per year. It's also important to work with different arrangement of dispense nozzles to create custom patterns. There are a variety of dispense technologies used for this reagent deposition, which I will talk about in detail. AirJet is used to deposit gold conjugate. BioJet is used to deposit antibodies. Frontline is also used to deposit antibodies. The difference is BioJet's non-contact, Frontline's contact. And for those aforementioned microarrays, Piezo dispensing technology can be used for those smaller volume drops. The AirJet is a syringe pump driven aerosolizer which is used for depositing gold conjugate onto fiberglass. It's syringe pump driven, making it highly accurate and precise. It's capable of operating over a wide volumetric range all the way up to 24 microliter per centimeter. The flow rate is controllable and adjustable through the adjustment of positive air, and the spray width is adjustable, making it possible to create a more dense or wider cone across the substrate. All of these things lend to a very consistent aerosol, as is shown here on the left, of AirJets in various arrangement, spraying gold conjugate onto fiberglass membrane. All of these dispensers that I'm discussing allow for independent control of dispense channels, which further lead to the lack or avoidance of cross-contamination. Here's an example of a custom arrangement of AirJets used for completely impregnating fiberglass with different gold conjugate. AirJets can also be used for impregnation of material for blocking or sample pretreatment. BioJets are also syringe pump driven, and they are used for the deposition of antibody testing control lines. Reagent is taken from a source bottle, guided through a syringe pump. Microstepping from the syringe pump delivers a set volume to a solenoid, which opens and closes based on a specified open time. This simultaneous coordination of microstepping and a microsolenoid firing creates the proper kinetic energy to break off a drop of liquid onto a substrate. This works over a large dispense range, anywhere from 2 nanoliters per drop to 2 microliters per drop, working in multiple modes. Typically, for lateral flow development, we will be looking at on the fly, meaning drops will be deposited to create a tight line, and motion and dispensing are coordinated without stopping. For certain applications, particularly arrays, step and repeat is also an option to create these patterns where motion is temporarily stopped between dispensing. As with the AirJet, there is an independent control over each dispense channel. And unlike the AirJet, because there is independent control over a syringe pump sitting in a microsolenoid setting, the user can adjust the dispenser for different fluid rheologies, and this is very critical for drop morphology. Reagent sources can be bulk, meaning they start from a container, or for R&D level work, the syringe pump can be reversed to aspirate from, for example, a microtiter plate. All of this lends itself to high dispense rates. Pictured on the right are 4 independent BioJet channels. These are the solenoids, these are the ceramic tips. Here is a video of 2 BioJet channels dispensing test and control line on 25 millimeter wide nitrocellulose. This is at a fairly moderate dispense speed. This is an example of on-the-fly dispensing, where drops are pitched tightly to create concise lines. On the right is an example of arrays and lateral flow. 3 different BioJet channels dispensing arrays on the fly of 20 nanoliter drops. The speed shown here is 30 millimeters per second. However, the speed can be run at a more vigorous 60 millimeters per second, which is shown here. Each one of these arrays would represent a separate test. Frontline technology is another type of technology used to deposit antibodies. Unlike the BioJet dispensers, Frontline is contact, using flexible tips made of PEEK material. Syringe pump delivers a quantity of volume in contact with the membrane, and the tips drag or glide across the nitrocellulose. For more specialized applications, piezo dispensing can be used, for example, to create multiplex lateral flow arrays. These include arrays that are sometimes quantitative for a test, or perhaps tests which otherwise would be ambiguous. Piezo dispensing works over picoliter to low nanoliter range for drop dispensed, driven by a piezo element. This is typically used to aspirate, but also can be bulk fed for certain larger scale operations. High density arrays can be created as shown here. And just because this is piezo does not mean throughput is sacrificed. Some advantages from this type of dispensing, first of all, are the ability to create geometric symbols, alphanumeric symbols, and this tends to lend itself to better quantification, higher sensitivity, while lowering reagent use. In order to create these specialized arrays, software and an easy-to-use graphical user interface is often required. A pattern can be easily created, which then leads to the creation of these symbols, such as the one shown on the right. When considering dispensing of reagents, it's also important to consider quality control during dispensing. Quality control often requires some type of vision application. This can be achieved using a camera to look at membranes during the dispensing process, as well as a bad part marker to use some type of feedback mechanism to blot out any section of membrane to be discarded later. For dispensing, these vision applications are often used to look at line, position, width, and integrity, as shown here. This is critically important because poor line integrity can lead to ambiguous results or false negatives, but also line position can lead to bleeding of reagents or the excise or overlay of that line during lamination. This is also very critically important for array-based applications that require a reader window to fit in. This type of vision application can also be used for conjugate, primarily looking at conjugate width, position, and even concentration. Besides dispensing, vision QC is also important during lamination, primarily for position and overlap, because if the overlap is too great, for example, in building up a lateral flow device, a test line can be lost or a control line can be lost. And when considering reagent deposition and vision and quality control, it's important to put them in context of batch production and inline production. For batch production, materials are typically pre-cut to length. Membrane can be pre-laminated prior to reagent deposition, or the individual layers can be processed before lamination. Batch production usually is in the context of R&D to low-level production, typically less than 1 million tests per year developed. Quality control, while sometimes automated, is usually manual. This is a picture of a batch-level system for reagent deposition, capable of depositing antibodies on nitrocellulose and spraying gold conjugate through an AirJet. Any sample pad pretreatment or blocking, and sometimes even conjugate impregnation is accomplished using some type of tray for dipping, and all materials are dried in an oven. Drying is typically the rate-limiting step. A small laminator is used to assemble the test to make layers, as shown to the left, and a guillotine cutter is used to cut strips to the desired length. Often the goal is to start in batch level manufacturing, and once a process is set, to move to larger scale production. However, there are many things to consider in this transition. First of all, material selection. Oftentimes, and this is true in COVID-19 testing, certain materials are chosen for higher sensitivity results. However, these materials are often brittle, thin, and sometimes have poor tensile strength. When considering inline manufacturing, sometimes 100-meter rolls of materials need to be tracked. So tensile strength is very important, in addition for web handling and tracking. The design process should transfer easily to manufacturing, not just from a dispensing perspective and a time perspective, but also from a process perspective. For example, transitioning time from lot to lot or batch to batch or even application to application. And drying. Drying, again, is the rate limiting step. In the batch level manufacturing, materials are often dried overnight. However, inline manufacturing, drying needs to be faster. It's also important to use equipment that can be transitioned easily from batch to inline manufacturing. Fortunately, the dispensers that I discussed earlier do just that. In looking at inline high-level manufacturing, materials are continuously fed from a payout module to a take-up module. This is typically using the on-the-fly dispensing that I mentioned earlier. This provides for great process control, including continuous drying, inline QC with vision, and precision alignment due to tracking. Here we see a series of syringe pumps. These are tandem, meaning 2 pumps feed 1 line. This allows for continuous dispensing while one syringe is emptying, the other one is filling. That way the process never has to end. This is in the context of higher volume production capabilities. For example, at 50 millimeters per second, 100 meters can be treated in about 34 minutes. This is enough to produce 20,000 tests per year. Built-in environmental controls are possible, both in the dispense module as pictured here, also to the take-up module. The whole goal of this, besides increasing throughput, is to reduce waste and to reduce QC time. Here is an example of an inline system for dispensing and drying. A system like this is capable of producing up to millions and tens of millions of tests per year. A roll of material is fed and driven through a series of dispensers. The dispensing is analyzed via QC, potentially with feedback. Oftentimes, impregnation for blocking is included. Material is fed through forced air dry towers and then rolled up on what we call a take-up. In addition to the features I just mentioned on the other slide, other features can be added depending on application. For example, for poor tensile strength material, ultra-low tension options are available, which reduce inertial force on the membrane. Humidity control enclosures can be included around dispensing and take-up module. And for many applications where moisture is critical, a moisture sensor can be added inline. A newer trend in inline manufacturing is PLC systems. in order for proper control, monitor, and communication between each of these modules. Not only does this allow for everything to be controlled more centrally, for example, this system to this system, but also for more proper data collection and feedback. Here is a video of a reel-to-reel 120 feeding nitrocellulose membrane, as shown here, being driven through a system of BioJets dispensing 5 antibody lines. These tandem pumps ensure that the process is never stopped. Now this is nitrocellulose. For conjugate, a separate process would be used on the same system where fiberglass would be fed to that AirJet. Here you see 5 lines on nitrocellulose being analyzed with inline vision and a bad part marker before being dried and rolled up in the take-up module. Following inline reagent deposition, the layers are assembled into a test in a process called lamination. Auto-lamination includes web speeds up to 100 millimeters per second, which is highly dependent upon web material being run as well as the number of laminates or how complicated the test is. This process is designed for quick alignment and changeover. That way, multiple products can be run on a single system. The system is modular, it's flexible, it can be run with multiple applications, and these modular units can be added and subtracted from the system later as needed. Typically, after lamination, the finished layers are fed to a cutter for cutting. These can be first fed to MasterCards in a reel to be transferred or cut inline. Here's an example of an auto laminator. From left to right, a module laying backing card down before being laminated with nitrocellulose containing antibody test and control lines, then having a conjugate pad added. And these layers can then be analyzed with vision for overlap and precision before being laid with cover tape or barcode. In this scenario, either the tests are cut in a guillotine cutter or rolled up for a downstream process. Here is a video of an auto laminator. The backing material with a take-up liner, the membrane payout with tracking and take-up liner, and 3 material layout in this case, including conjugate. The vision inspection, the cover tape payout and placement before the cutter, cap stand for tracking and driving, and the take-up reel. In addition to basic lamination, barcode labels can be added for the test and for vision inspection recognition. After lamination, cards can be collected and stored instead of cutting, or the final assembled test can be rolled up. The last process prior to bottling or packaging is cutting. These are a couple of the cutting options besides the guillotine cutter. In this scenario, a rolled up test can be fed to a cutter to create specific lengths. These cutters can be used with various sensors, including a target sensor for fiducials. In this picture, a bottle collector is added to the end of the cutter to collect the cut test strips. Here, shown is a cutter that is used in batch level manufacturing. Pre-cut cards that are laminated are fed to a cutter and then bottled. In this picture, A rolled up laminated test is being fed to a cutter, which then sorts into 2 different bottles. Besides the standard lateral flow test that contain test and control lines, it is possible to create multiplexed arrays and geometric symbols. Besides multiplexing, this can lead to more sensitive quantitative tests, but also qualitative tests to eliminate the possibility for any ambiguity or create a certain message. Multiplexing can be done at the batch level or inline web handling in a high throughput manner. Oftentimes this is continuous. However, for certain complicated arrays, indexing is important at a high speed that enables multiple arrays per second to be created. Reagent handling is critical. In particular, oftentimes higher concentrations or more viscous samples are required. Typically, this process is going to be using picoliters to nanoliters as opposed to microliters, as shown here in 50 nanoliter spots on nitrocellulose. Positional accuracy is very critical for this, particularly for multiplexed arrays that require some type of reader. For this reason, fiducials can be used often with a vision system, and software is used for editing an array, which is particularly ideal when looking at R&D level manufacturing. Here is an example of batch level multiplex arraying, which include pre-cut card, various dispense channels, dispensing an array created through a vision program. This process can also be adapted to inline manufacturing, oftentimes with arraying. For example, here is a schematic or a concept of material being fed into 2 separate dispense modules for indexing to create these more complex arrays before being dried. This concept also shows inline lamination and cutting and bottling, which would be the ideal process. Oftentimes, customers choose to separate these out. That way, multiple lots, in addition to multiple tests, can be developed. When looking at multiplexed arrays, particularly in smaller volumes, QC is especially important for inline manufacturing because it's critical to know exactly when to make a cut and when to dispense arrays. For this reason, fiducial markings are also used. These fiducial markings not only allow a cutter to trigger cuts as needed down the process, as well as lamination, but can also be used to trigger dispensers to deposit a very specific pattern within a certain window. Thank you for your time.
And thank you, Dr. Fronczek, for that outstanding presentation. We will now move into the live Q&A portion of the presentation. And as a reminder, please submit your questions in the Q&A box. Okay, our first question is, can you give some specific examples of arrayed LFDs?
So arrayed LFDs are not nearly as common as the standard LFDs striped with single lines. However, typically we'll see multiplex tests that require a higher level of sensitivity that can be matched up with some sort of a reader. And one of the big advantages of the arrayed is that the customer can use less reagent and also save costs on the test. In particular, I have worked with a couple of these type of arrayed LFDs in the past that deal with the detection of certain toxins such as anthrax.
Wow, okay, thank you for that. And the next question that we have is what has been the biggest challenges when manufacturing COVID-19 tests?
So the nice thing about our dispensing technology is that going from different types of tests, different types of biochemistries is often directly translatable, since we can kind of tune for different fluid rheologies and properties. In all reality, the biggest challenge that we faced is customers who are pretty new to this arena, to LFD type of tests, jumping into the COVID arena with some of them little experience. The other part of that will be a lot of customers who have been more in R&D type of manufacturing, jumping to inline for COVID testing. So it's mainly been assisting customers kind of making that jump in a very short period of time, which has been very common in the last 8 months, as you can imagine.
Thank you so much, Dr. Fronczek. And I have a few more questions here. How many tests per year are possible when batch manufacturing?
Typically, and this is really heavily dependent upon the actual process, we have seen a single system manufacturing in the low to mid hundreds of thousands of tests per year. In order to really get closer to the million tests per year, it usually requires maybe 3 shifts a day and often multiple pieces of equipment. And at some point, it usually makes more sense to just scale to the inline type of equipment for that. So usually it's going to be in the low to mid hundreds of thousands, maybe up to about 500,000.
Thank you so much. And our fourth question today is, what is the most challenging LFA application BioDot has experienced?
So typically it's going to be one of 2 scenarios. Number one, it's often pretty common to select certain materials, particularly very thin, unbagged nitrocellulose, which is often good for higher sensitive type of test. Transitioning from batch to inline, where web handling is critical, using those type of materials with tensile strength is challenging. The other challenge is, and really kind of what I mentioned in the talk, the time limiting step is drying, particularly when depositing a high volume of reagent. So often a challenge is to find a scenario where we can keep a very high throughput, but also making sure materials are tracked properly, materials do not break in line, and materials are dried before they're assembled in a single test.
Thank you so much. And our last 2 questions here is, what is typically the biggest challenge when transitioning from batch to inline manufacturing?
Pretty much the answer I just gave. It's mainly drying, but really developing the process around continuously processing tests for an hour or so, as opposed to doing batches. It's really adapting a process and making sure that when developing a batch process, it's easily translatable to inline without having to go and reinvent the wheel.
Wonderful. Okay, and our final question today is, how common, Dr. Fronczek, is it to combine dispensing, laminating, and cutting into a single inline process?
The answer is it's not very common. Usually lamination and cutting are combined. The problem, again, usually is drying sufficiently, particularly when running at a higher speed for good throughput. The other thing is that a lot of times customers are running multiple types of tests, so it doesn't make sense to have everything in line. That way they can easily switch over to a test to dispense, and then at the same time they can be laminating and cutting for another type of test, if that kind of makes sense. So usually it's not very efficient. Plus, an inline system that combines all three would be very long. So it would require quite a bit of space. Usually, instead of having a nice, long process like that, it usually makes sense to stack systems side by side for space purposes.
Thank you again, Dr. Fronczek. Unfortunately, we are out of time for today. If you have any questions that were submitted and not answered today by our speaker, will be addressed via email. And thank you for your outstanding questions to the audience. And the presentation will be available on demand for viewing for 12 months. Please remember to share it with colleagues who might be interested in today's topic. And don't miss out on other presentations on our agenda. Visit the agenda tab in the auditorium for a full listing. Thank you again for your participation, and until next time.
Thank you.